Hi, everyone. Good morning, good afternoon, and good evening, wherever you are. Thank you so much for joining us today. And I see the numbers are at around 175 people. This is absolutely terrific, and it's still growing. So we'll let people join us as we go, and I'm confident that we are going to have a really good number of participants from across the 5 Neuroscience Consortium, all the partners and stakeholders that we work with across the Neuroscience Consortium within the Critical Path Institute. So my name is Sudhir Sokmaran. I'm the Vice President of Neuroscience and also the Executive Director for the Critical Path for Alzheimer's Disease Consortium at the Critical Path Institute. I'm very excited for this first Neuroscience Program webinar that we are hosting. And this is going to be the first in a series of webinars we will have as we go forward at some reasonably timed intervals on some key topics that are relevant to our efforts and relevant to the efforts of the industry drug development teams across all the 5 neuroscience therapeutic areas that we work in. So this first webinar is on neurofilament light chain and neurodegenerative diseases. We'll have a presentation by Dr. Terina Martinez, who is the Executive Director of the Duchenne Regulatory Science Consortium and for the Critical Path for Therapeutics in Ataxias. After that, we will have a panel session, which participation from some experts in the space who are going to be joining us, and we thank them so much for their time and availability. So just a quick logistical items. If you can all please remain on mute, we will unmute you after the presentation from Terina, and we will be taking questions from all of you and any of you. Feel free to raise your hand on WebEx or please put in a comment on the WebEx chat. We have a team internally who is monitoring the WebEx chat, and we'll be channeling those questions to Terina during the Q&A. We will make sure that we address and answer any and all questions that come through. And of course, if we run out of time at the end of this webinar, we'll make sure to capture those questions and bring it to one of the upcoming coordinating committee calls. So with that, I welcome you all once again. Thank you so much for your enthusiasm and participation. I will hand it over to Terina to get started with the presentation. So Terina, if you're ready to go, it's over to you.
Thank you, Sudhir. Thank you for the introduction. And again, thank you all for joining us today. It does seem to be a topic of interest. This has, of course, been a topic that has been very much on the minds of all of the scientists and researchers at CPATH as we have been having these conversations in smaller micro sessions within the individual consortia. So we thought it'd be advantageous to bring it into the bigger picture and to share the learnings across the different diseases. So with that, I will kick off. As Sudhir mentioned, I'll just do a quick run through of some background and level setting case studies, and then we'll start off with the panel discussion. So just the quick outline, I'm going to start off with a review of neurofilament light chain, its molecular mechanism, and the biological significance that it has as a biomarker. I'll go through some assays and clinical validity questions across different neurodegenerative diseases. I'll also discuss validation case studies across many diseases with an emphasis on clinical trial readouts that have used neurofilament measurements. I'll also go through and discuss and set up for the panel discussion the current challenges and unaddressed questions for neurofilament light as a clinical trial-ready biomarker. And then we'll also end up with some future directions for programs aimed at advancing neurofilament light as a fluid biomarker for neurodegenerative disease research in a manner that is maximized for regulatory success. And then we'll transition into the panel. I'm going to go ahead and stop sharing my video just so that the bandwidth is a little bit better and that there aren't any glitches as we move through the presentation. So just want to begin with kind of going through what I consider to be an attempt at deciphering the complexity of neurofilament light and really the biomarker landscape in general in neurodegenerative disease. This is just a bit of an analogy, looking at an iceberg with the idea that what you see above the water is absolutely not representative of the whole size, complexity, and breadth of the iceberg. And the quadrants that are split up here on the screen, the known knowns and the unknown unknowns, for example, really relate back to a Pentagon press briefing back in the early 2000s with General Rumsfeld talking about the war in Iraq and the known knowns and the unknown unknowns. And then the unknown knowns quadrant was recently added by a Slovenian philosopher to just really cover what would be, the main quadrants in a very complex situation. So relating this iceberg analogy and the complexity to neurofilament, in the known knowns category, we know that neurofilament is a priority fluid biomarker candidate. It has actually had much more robustness than many other fluid biomarkers for neurodegenerative disease, but we also know that it's not specific. When we look at the known unknowns, we know that while NFL has potential, we don't really have a completely clear picture of its dynamics and what it means across several neurodegenerative diseases longitudinally. When we look at the unknown unknowns, I'm putting in this category the really big black box of neurofilament's performance in clinical trials. I would say that until we get a clearer picture of the performance of the biomarker in neurodegenerative disease in just natural history studies, we really can't get into the complexity of the clinical trial space, although some diseases are positioning themselves there, but others are still a little bit behind. And then when I think about the unknown knowns, which again is a little bit of a brain teaser category, here I'm really focusing on the fact that different diseases have investigated neurofilament either longer or in many more sites or studies with higher power. And there will be an opportunity, I believe, through this conversation, through the panel discussion, for what is unknown in many different neurodegenerative diseases to really begin to learn what is known about the biomarker from other diseases. So these would be unknown knowns. So with that, I hope I've level set that, of course, neurofilament as a topic and as a biomarker for neurodegenerative disease is complex, and we will work iteratively through some case studies and discussing analytical and clinical validity to really understand and better tease out some of this complexity. So looking at the molecular mechanisms and the biology of neurofilament, this is a diagram that really captures how it is amenable as a biomarker. Neurofilaments are abundant neuronal scaffolding proteins, and specifically there are several subunits of neurofilament, and the one that is the most focused upon for biomarker applications is neurofilament light chain. Throughout this presentation, I will abbreviate neurofilament light as NFL. It is highly specific for neuroaxonal damage, but what's important is that anything that causes the damage will cause the release of the neurofilament, but this is agnostic to the primary neuronal damage trigger. This is why it is a very specific biomarker for neurodegenerative disease, even in fluid-based matrices. But it is not specific for individual diseases because whatever the insult is that leads to the axonal damage will lead to the signal increase. It is detectable in both cerebrospinal fluid and blood. The concentrations in CSF are higher than in blood, but the correlation of the amount is very, very close between CSF and blood. The initial first and second generation assays for neurofilament detected it in serum. These were immunoblot or dot blot or sandwich ELISAs. But as the assay technology improved, moving on to the third and fourth gen assays, either enhanced chemiluminescence or SIMOA, single molecular SIMOA, those assays are sensitive enough to detect the lower levels of neurofilament in blood. And so with whatever the upstream signal is, the neurofilaments get broken down and degenerated into the CSF, into the blood. The mechanism of how this transfer occurs is not completely understood, but once it's in the CSF or blood, it's very stable. So looking then at the underlying principle that neurofilament is a specific marker for neuroaxonal damage, but it is not specific to the different types of neurodegenerative disease is further illustrated in this slide. Selective neuronal vulnerability and brain region specificity and the progressive pathology that underlie the complex etiology of neurodegenerative disease is what does lead to the clinical manifestations that are reflected by the brain region and specific cell populations that are affected. And that's depicted in the diagram. So whether you're looking at dopaminergic neurons in the midbrain in Parkinson's disease, or medium spiny neurons within the striatum for Huntington's, or motor neurons, for ALS, the different neuronal sensitivities and vulnerabilities will lead to selective degeneration of that subset of neurons, leading to then the clinical manifestation. So the neurofilament will pick up the signal of any of these discrete neuronal populations as they degenerate. Of course, it's also going to pick up other signals if there's, for example, acute injury like stroke or ischemia, brain injury. It's really going to detect any insult or injury to the neurons. But I think what's important is to understand that within every individual neurodegenerative disease, the unique trajectory, the progression, and the mechanisms that contribute to the etiology are very complex and do impinge directly on the neuron populations. And we can begin to get clues from about what's happening in these discrete and complex neuron populations. So while NFL is not disease specific, when we look at it over time in these individual diseases, we can begin to get some information about what's happening and can certainly leverage it as a biomarker across many of these neurodegenerative diseases. So just coming back briefly to the iceberg analogy, this is a really nice figure that I pulled out of a recent review article that is highlighting the longitudinal trajectories of serum neurofilament light. Specifically, it's highlighting the potential differences in the natural history of various neurodegenerative diseases, indicating that there are variable durations in the pre-symptomatic phase. The idea is that depending on the disease, there's either a very long or a moderate or shorter period of time where the neurons are degenerating and the neurofilament is being released into the CSF and blood, but the manifestation of the disease has not occurred. So this is in the prodromal or pre-manifest stage. Once a certain threshold of neurodegeneration occurs at the clinical threshold, then there's a phenoconversion and frank symptomology is observed. So there have been many studies across these diseases that show that for AD, for example, the period of preclinical onset, before clinical onset in that pre-clinical phase, neurofilament is detectable. And for MS, it's also detectable, but not quite as far out as AD. The more acute or faster progressing neurodegenerative diseases like frontotemporal dementia or ALS also have detection in this preclinical phase, but the rate of progression is faster, the slopes are a bit steeper, and then they move into to the manifesting or clinical threshold. So the duration of the preclinical phase could be a characteristic that depends on both the neurological reserve of the subject and the specific pathological process that's taking place in the CNS. So shifting gears specifically to how to define and really understand the specifics of neurofilament within the broader biomarker category and complexity, this is just a graph that breaks down the various different biomarker categories, ranging from diagnostic, monitoring, predictive, prognostic, pharmacodynamic and response, safety and susceptibility. The ones that we're really going to focus most on today are a couple of instances of using neurofilament as a companion diagnostic. So by itself, it won't be diagnostic for a specific neurodegenerative disease because it's not specific, but in partner or in companion with other disease-specific markers, it can be useful. It's used and reported broadly as a prognostic biomarker, and it can also be predictive as well. And we're also going to look at some clinical trials that are assessing utility and potential for pharmacodynamic response. There are also some additional terminologies that are often discussed in the literature, put into papers, presentations like this, that discuss the biomarkers in these non-specific ways that are really broken down for preclinical and clinical and drug development work. And that's using, additional terminology like exploratory. But again, if this is a biomarker that hasn't been firmly identified and fully vetted through regulatory approval, then it is an exploratory biomarker. I mentioned that for purposes of diagnostic, it can be a companion. Obviously, there's, questions of differentiating biomarkers, recurrence for situations like oncology. So many of these terms will also come into play. But for the most part, I am going to try, as we go through the case studies, to link the various indication from the case study to these specific biomarker categories. keeping in mind that it's the utility of a prognostic biomarker, for example, in its application to stratify patients to enrich in clinical trials based on particular inclusion or exclusion criteria, that is really valuable. And of course, a response biomarker would be key because it would indicate efficacy or show trajectory of the effect of a test drug. So in order to really evaluate the potential for neurofilament as a biomarker for neurodegenerative disease, we really do need to look at the considerations of analytical validity and clinical validity. So the analytical validity is focused on the assay performance. This really does consider pre-analytical factors, also assay standardization and data analysis and reporting. All of these need to be standardized and applicable across sites and centers. For clinical validity, the neurofilament performance as a surrogate of disease-related clinical outcomes is really the key. And in this case, there needs to be a biological rationale and also strong supporting data. We've discussed before now the molecular and biological rationale for neurofilament as a marker for neurodegenerative disease. And then we'll go through now some case studies and a bit of an assessment of the supporting data. It's important to note that for regulatory approval for biomarkers, both clinical validity and analytical validity have to be met in order for it to move forward. So focusing now on specifically analytical validity, we're not going to spend too much time here because by now there's been a wide body of literature across multiple neurodegenerative diseases that have assessed analytical validity, but I do think it's important to just take a moment to take a look at that. On the left, we're looking at an example of the correlation coefficients for CSF and blood across 3 different types of assays, either the ELISA, the enhanced chemiluminescence, or the Simoa. And in this case, for multiple sclerosis, you can see that that the Simoa assay performs better in terms of correlation coefficient and approximates 1. So I'd say that now really the status of the field, especially if you're looking in blood, whether plasma or serum, the Simoa assay is truly the most sensitive, and that has been conserved through many different publications. And it's not just something that's unique to MS. This is just a snapshot of a couple of papers that show the assay validity on the top first for Parkinson's disease, on the bottom for Huntington's disease. The paper on the top was looking at blood-based neurofilament light as a biomarker for differential diagnosis of Parkinsonian disorder. In this case, they looked at NFL in blood and correlated that to CSF NFL and showed that when they measure it in blood, that it can discriminate Parkinson's disease from atypical Parkinsonian disorders. And then in the paper at the bottom that's showing the example of analytical validity for Huntington's disease, this was a study that evaluated neurofilament in CSF and blood in parallel with clinical evaluation and MRI in pre-manifest and manifest Huntington's disease mutation carriers. So they enriched the study for carriers of mutant Huntington and were able to assess the NFL levels both in serum and blood. And so this shows just the general consensus that there is analytical validity for NFL. I've only shown you these snippets for multiple sclerosis, Parkinson's disease, and Huntington's disease, but for other neurodegenerative diseases that are being presented in the case studies upcoming, an analytical validity will also come through. So this is just looking at a case study of meta-analysis for neurofilament as a differentiation marker. So again, we know that it's not specific for one individual disease, but it can be utilized to differentiate certain diseases from other diseases. In this particular case, it's a meta-analysis looking at a forest plot of serum neurofilament light, comparing frontal temporal dementia, PPD, which is other primary psychiatric disorders, and healthy controls. It turns out that there's a really high rate of misdiagnosis between PPD and the behavioral variant of frontal temporal dementia. And this can, of course, change the course of treatment, have many important downstream effects. And with such a high rate of misdiagnosis, it's important to have a biomarker that can differentiate And since there are structural functional imaging markers lacking for early FTD, NFL was studied as an opportunity to differentiate between these. So you can see, just looking at the graph, that at the top, looking at the PPD condition, in the middle of the graph is the FTD condition. At the bottom is the healthy controls. The subjects with primary psychiatric disorder align close to the levels of neurofilament that is observed in the controls. But in all the cases of the frontal temporal dementia, the NFL levels are higher. So even though the clinical manifestation may be overlapping and unclear initially at presentation between these PPD and FTD patients, looking at the serum neurofilament can differentiate between those. So this just provides a case study indicating that FTD has utility in certain situations as a differentiation marker. This is a summary from the ALS biomarker cross-disease dashboard. This is really bringing together a really powerful tool to compile all of the different data and provide an opportunity for meta-analyses across neurodegenerative diseases and different biomarkers. In this particular case, I'm focusing in specifically on markers for neuronal injury, and this covers a wide variety of neurodegenerative disorders across the top, whether they're the amyloidogenic, tauopathies, synucleinopathies, prion disease, or others, which include depression, vascular dementia, etc. So looking at the heat map, what's important to note here is that this is a comparison back to Alzheimer's disease levels of neurofilament. So an equivalent neurofilament level to Alzheimer's disease would have a score of 1 or a value of 1. Anything lower would be in blue colors. These would be lower levels than Alzheimer's disease. Anything higher would be in the warmer red colors. If something is in bold text, it means that the p-value is less than 0.0001. And so looking at this, this summary here, looking at NFL, CSF in this top line, and plasma and serum NFL in the bottom line, clearly not every disease is represented. There's much work that still needs to be done to analyze NFL in this space. But you can see for certain diseases like ALS and Creutzfeldt-Jakob that the neurofilament levels are very high, much higher than in Alzheimer's. In some of these other tauopathies and synucleinopathies like MSA, it's also higher. But in particularly in Parkinson's disease, there's a conserved indication that CSF and NFL and CSF and serum are lower than in Alzheimer's, and in Parkinson's disease dementia as well in the studies that are captured within this meta-analysis. So we'll dig into some of these case studies that contributed to this data in just a moment. But before we do, I want to just go through a couple more meta-analyses of from this biomarker platform. So this is looking at Alzheimer's disease versus control, specifically looking at neurofilament in CSF. I want to point out that this is using an ELISA assay. And this is looking at a bunch of different studies over many decades now, assessing neurofilament in AD versus control subjects. This forest plot is showing the effect size, and in this case, effect size is going to be the amount of a larger effect size is going to indicate a stronger relationship between the two variables. So the z-score is the significance, and that's also shown. And if you look at this particular graph, you can see that the effect sizes for neurofilament, even all the way back into the mid-1990s, hover in the range of 2. So this is 2 standard deviations greater than the mean, and this shows that this is a conserved and significant effect. Looking then at Alzheimer's disease versus control, again, neurofilament, this time plasma and serum, you'll see that this is shifting to more recent years because the sensitivities of the Simoa assay and other assays allowed for detection in blood. And again, looking at either the Simoa, the immunomagnetic reduction, or ELISA by color, there's still an indication that the effect size is around 2. And I think that this again shows that now that we're in serum, both NFL and serum can detect the NFL in CSF and serum, and the effect size is conserved around 2. So this is another case study meta-analysis graph. This time we're filtering for NFL, and you can again see the both in AD versus control CSF in the gold color, and AD versus control in plasma and serum in the purple. It's again at an effect size of 2. Now adding in A beta, so with whether A beta 40 or A beta 42, you can see that none of those really get above 1. A score of 1 is equivalent to between the control and AD, and below that would be, of course, less. So again, neurofilament is staying above at an effect size of 2, but both of the versions of amyloid beta do not get above 1. Now adding in tau, we do see an increase in tau. In some of these reported papers and studies, different versions of tau have been shown to have high effect size. Specifically, the 3 that are kind of up here, the top of the left, the highest effect sizes represent either tau, phospho-tau 217 or phospho 231. And then total tau is the largest gold dot on the side. But you can see that when it comes to differentiating AD versus control, except for a few phosphorylated versions of tau, NFL is really robust and more effective than amyloid beta. So then just one more graph of heat maps, and then we'll go into a couple of other different case studies. This is a particularly interesting report that did a really thorough investigation of diagnostic accuracy of plasma neurofilament light in neurodegeneration. This was a multi-center validation study looking at the diagnostic accuracy across neurodegenerative diseases. Again, the area under the curve is being shown by the heat map for the warmer colors having a higher level and accuracy than the blue. And the summary here is that the plasma neurofilament light is clinically useful in differentiating across certain types of the diseases. So not every disease that's being looked at here is showing an effect, but it can certainly tell, for example, the difference between atypical PD and regular PD. It can identify dementia and Down syndrome. And it can distinguish neurodegenerative disorders from depressive orders because those don't tend to show a high correlation or a high level of neurofilament. So really what I think is important here is that the study was looking at 2 very large cohorts and showed assay harmonization and conserved to the extent that the different subjects were matched, or in terms of disease cohorts, subcohorts, they were represented in both cohorts. It showed, highly conserved effects. It also pointed out the importance of age-related cutoffs because neurofilament does increase with age, and the additionally comorbidities are going to increase with age, which could also cause higher baseline levels of neurofilament. And so normative analysis needs to happen, and cutoffs need to be really thoughtfully applied to different studies in order to account for the age-related neurofilament effects. Moving from the iceberg analogy now to the other horizon of space, just really kind of thinking back to how we can link biomarkers to clinical outcomes, and the fact that for neurodegenerative disease clinical trials, this really is a moonshot. It's incredibly challenging. Everyone, I think, participating in this session will appreciate the difficulty and complexity of linking anything to clinical signs and clinically meaningful endpoints in neurodegenerative disease clinical trials, whether that's a imaging biomarker, a fluid biomarker, a scale of some sort. It's very challenging to get these to link up. And so having a biomarker that has utility as a diagnostic tool, which is what we focused on for neurofilament up to now, is useful. But in order for it to have maximum utility in clinical trials, it needs to be sensitive enough to reflect disease progression and or severity. So that's the moonshot we're talking about. And we're now going to go through a few clinical trials to see what the status is for NFL in its position as a potential biomarker in neurodegenerative disease clinical trials. So this is just first a layout of a study for NFL, a case study in NFL and inherited Alzheimer's disease So in this case, they were looking at serum neurofilament and its ability to predict neurodegeneration and clinical progression in pre-symptomatic Alzheimer's disease. This specifically was a cohort from the dominantly inherited Alzheimer's network out of the NIA. It was a longitudinal study that looked at serum NFL, highly powered, and it showed that it can discriminate between mutation carriers and non-mutation carriers. And it peaked at the point of phenoconversion from pre-symptomatic to the symptomatic stage. They showed high correlation between CSF and blood levels of NFL, and also between NFL levels in blood and MRI, which quantified cortical thickness and cognitive performance using either the Mini-Mental Status Exam or the logical memory test. And so this study looked at it determined the predictive potential for neurofilament as a clinically useful biomarker for disease progression and neurodegeneration in genetically inherited Alzheimer's disease by comparing neurofilament levels at baseline over time, and also looking at these additional comparators. So this is just a quick snapshot of one of the figures from that paper that I just outlined, looking first at baseline on the top, and then a follow-up at 18 months at the bottom. On the x-axis, we're looking at either the rate and change of MRI, the rate of change in Mini-Mental Status Exam, or the rate of change in the logical memory test. All of the y-axes are showing the rate of change of serum neurofilament in a log scale. And you can see from this study that the conclusions that we outlined in the previous slide support that NFL has utility as a progression biomarker because it can predict eventual neurodegenerative effects in asymptomatic individuals. And I think that this is, a good example of utilizing that obviously long and many years of pre-symptomatic phase that Alzheimer's disease has. When we think back to the earlier figure that demonstrated the long slope of neurofilament for several years before clinical manifestation, this is a specific and tangible example of that phase. But importantly, this is leveraging the fact that the genetic diagnosis can be utilized in order to stratify and differentiate between these subgroups. Looking now at another case study, this time neurofilament and Parkinson's disease. You might remember from the previous heat map I showed from the ALS Forum biomarker dashboard that a lot of times neurofilament levels in Parkinson's disease are lower than Alzheimer's. But that didn't show, of course, the difference between neurofilament within a Parkinson's disease cohort looking at Parkinson's levels of neurofilament versus healthy control. These are just two example case studies. The top one looking at CSF in serum or in CSF or serum neurofilament light in combination with alpha-synuclein as a biomarker panel to discriminate Parkinson's disease from controls. And then on the bottom, it's looking at individual neurofilament dynamics in serum that mark the conversion to sporadic Parkinson's disease. So now, again, we're getting at the more defined level of granularity, not able to use genetic predisposition as a way to identify early manifesting or non-manifesting prior to phenoconversion. So this shows that in certain studies that NFL can be utilized to detect either in combination with other markers or in phenoconversion in sporadic populations. NFL can be leveraged to make these determinations. And I think that it's important to note that that these studies are looking at idiopathic populations. Often in the case for Alzheimer's and Parkinson's disease, idiopathic versions of the disease occur in later ages in life. And that, of course, is going to be in the time frame where neurofilament has increased due to age, and also when there are higher levels of comorbidity within subjects. And so this means that neurofilament still remains sensitive enough within that complexity and background to have utility as a diagnostic biomarker in companion and also as a prognostic biomarker. But we haven't really assessed or answered with these case studies the potential for neurofilament in progressive Parkinson's disease. And that is what is investigated on this next slide. This is a slide that is utilizing a couple of different cohorts of Parkinson's disease patients. One is the de novo cohort, which is a de novo cohort, and then also the PPMI cohort, which is the Parkinson's Progression Marker Initiative cohort that the Michael J. Fox Foundation has assembled. This is a landmark study that's an observational clinical study with many, many thousands of patients to assess longitudinally and determine and verify progression in Parkinson's disease. It contains genetic subsets, and it collects a wide range of clinical and biosample data, including imaging, DAT scans, many fluids, tissue samples, many different types of fluids and collections are taken, and the follow-up is carried on for years in many thousands of patients. So it has the opportunity to really begin to assess progression within Parkinson's disease. So in this study, they started off first with analyzing neurofilament levels in healthy control, PD, and other neurodegenerative disease. So this would have been patients that presented at the clinic with potential for Parkinson's disease, but then were determined at later clinical examination to not have Parkinson's disease. It was some other disease like MSA. And then they did, since this study was conducted using CSF and ELISA, a bridging cohort using CSF and serum analysis was performed. And then the analysis in the Samoa assay with serum in the PPMI cohort was done. And this is looking at healthy control and also prodromal, which would be a subset of the cohort that would have been identified with either REM sleep behavior disorder or hyponausea. And then they have their PD cohort, their genetic PD cohort, and other neurodegenerative disease. So this is, I think, an important study that does show that when you can look at some of these very specific differences between the subgroups, obviously, if you're looking, in the earlier stages, there's a significant amount of overlap, unless it's this other neurodegenerative disease group, which has high neurofilament. But it really is difficult to differentiate, for example, PD and some of the other groups within the sample. But what is interesting to look at is the slope and the continuing increase of neurofilament over time in this genetic PD population, because this is the one that is eventually phenoconverting as well. So I think that this is leveraging the strength of these cohorts and the fact that there's the ability to look at many subjects that have different subtypes and have high power. This does show that in very specific ways that NFL can be utilized for progression in PD, but that it's of narrow utility because it has to be, the levels and differentiation in Parkinson's disease are smaller, the deltas are smaller, and it also has to have the ability to follow the subjects longitudinally. But this did show that once age adjusted, longitudinal increases in serum neurofilament were higher in PD than in healthy control. And also that these changes in serum neurofilament correlated with the MDS UPDRS-3 score and some cognitive changes. So then focusing now on looking at a case study for NFL as a progression biomarker, this time switching to ALS, recognizing that ALS is much more aggressive in its progression, and wondering if NFL can be better utilized for progression in a disease that has a more rapid rate of advancement. So this is looking at longitudinal evaluation of neurofilament and inflammation markers in ALS patients using matched CSF and plasma samples from the Northeast ALS Biofluid Repository. They stratified the subsets into groups using the ALS Functional Rating Scale. And in this study, they demonstrated that there's longitudinal stability of both targets, the inflammation and neurofilament, and that neurofilaments have prognostic value for fast progression patients. So where I think that is the easiest to see, of course, looking at some of these box and whisker plots for NFL in CSF, NFL in plasma, the correlation between the two matrices is again conserved. And then when they look at a modeled progression, specifically focusing on the patients with Alzheimer's, that's the PALS, that are fast progressing, versus the ones who are slow progressing, you can see that looking at the levels of neurofilament on the x-axis, that the rapid progressing ones have the highest levels of neurofilament. And this is therefore indicative that in this particular situation of an ALS cohort, that levels of neurofilament are indicative of the faster progressors. Now looking at another neurodegenerative disease, specifically inherited ataxias and the subdivision of spinocerebellar ataxia 3. This is looking at associations of NFL with disease severity, disease progression, age, and CAG repeat length. And so what I think is important to note here, just briefly, is that SCA3 is the most common dominantly inherited form of neurodegenerative ataxia, and it is caused by expanded CAG repeat in the ataxin-3 gene. The irreversible motor deficit and decline begins in midlife. What I think is important to see on the left, we're looking at the cross-sectional assessment of serum NFL levels in 2 independent cohorts. So this is looking at a couple of different cohorts, which again, in a rare disease, is a high bar. And they're looking at using a homebrew neurofilament duplex Samoa assay. Using this assay, they determined intra-individual disease progression, which is key in a longitudinal study, and also they're looking cross-sectionally, and then they're also looking at NFL levels and how they significantly correlate with annual change, measuring it to the CERAS score, which is the scale for assessment and rating of ataxia. So this is linking this fluid biomarker to a clinically relevant scale. And in this study, they assessed association with age and CAG repeat length in the SCA3 carriers with a linear model using pooled NFL data from both cohorts, and found that both age and CAG length were associated with higher NFL levels. So this is, again, showing that, knowing that in SCA3, higher CAG causes earlier onset and more rapid progression. This, again, is conserving the takeaway that NFL is associated with those particular changes. And then on the right, looking at different CAG lengths and the different repeats, looking at over time, you can see that that within the intra-individual patients, that the correlation does maintain. So it's linear with age, and then the higher level of repeats are also associated with higher NFL levels. And so I think that altogether, this illustrates the potential for NFL as a biomarker for progression, disease severity, and patient stratification within SCA3. One more study looking at SCA3, again, linking it to the clinical scales. This is another cohort, prospective cohort enrolled in France. They used a neurological exam, MRI, and NFL at baseline, and at 2 years by the Quanterix Samoa assay. They found that NFL levels were significantly higher in SCA expansion carriers versus controls, and that age-adjusted NFL levels for all SCAs were significantly correlated at baseline with the CERAS score and cerebellar volume changes. So you can just see here that that the NFL levels are the ones that remain higher. Importantly, the NFL levels themselves don't appear to change much over time in the SCA patients, but other studies have actually seen changes over time. But it does support the use of NFL as a better biomarker for pre-manifesting spinocerebellar ataxia cases, rather than as an outcome measure in patients with overt disease for the short durations of time typical of a clinical trial, because this did assess a 2-year follow-up. So I'm going to move a little bit faster here so we can make sure we get to the panel discussion. I just want to cover a few case studies focusing on NFL readouts from clinical trials. So this is just a quick summary of NFL and its utility in clinical trials for MS. So MS is positioned to have more clinical trial use with neurofilament, because it has been shown to be associated with the different types of MS. And this is just a summary of some of the different clinical trials that have been assessed for MS, and looking at neurofilament. Just a quick glimpse at the correlations between baseline NFL and imaging outcomes are provided here. Sometimes it's not reported. Other times it is reported and associated with either changes in imaging or in baseline clinical outcomes. And so what I think is important about this particular review is that it pulled together all of these different MS clinical trials, demonstrated the robust analytical validity and clinical validity that NFL can have in MS clinical trials by relating them to imaging and disability measures, and therefore positioning it for potential responsive biomarker utility. They also did, of course, discuss some of the limitations of NFL because the technical challenges, non-specificity, and effects of comorbidities remain a challenge. This is just another quick summary table of the response of neurofilament light concentrations to treatment in progressive MS. So again, this is taking some more of these MS clinical trials. I'm not going to go through the details because they are varied. Some of these trials, again, had no effect. So there weren't really differences in NFL between the on-drug group and the placebo group because there was no effect. And then other times, of course, they're really trying to understand and assess longitudinally. But this is a nice summary of all of these different clinical trials using NFL in MS and the various outcomes and considerations that they considered. One last case study looking at Alzheimer's disease in NFL in clinical trials. This was actually a retrospective study that was done several years after the particular clinical trial looking at terinflubril for mild AD was assessed. This was looking at NFL and total tau in this 18-month interventional study utilizing these particular scores. And this was, many patients, 236 of the 500 were on drug, but no adjustment for treatment arm was done because the trial outcome was negative. So NFL was assessed in all of the different subjects and was found actually to have a stronger association with clinical tests than with total tau, and than total tau had with the clinical tests. And when adjusted for patient age, neurofilament still predicted future changes 3 to 6 months prior to the manifested changes on the clinical scales. So what I think is important in understanding this is that while it didn't cover and show significant changes in the neurofilament levels between baseline and at the end of the 18-month study, the neurofilament was differentiating for the early 3 to 6 month range, and this could be potentially leveraged to look at independent predictability and also may improve the ability and the capacity to identify changes in the rate of cognitive decline, and have therefore use as a response marker, and could also potentially be used to identify and enrich clinical trial populations for patients that are more likely to decline over a shorter period of time to allow a better opportunity of observation of drug change based on these neurofilament levels. So one last clinical trial case study is just a very recent one that's kind of hot off the press. And we're deviating a bit here, not looking at any of these age-related neurodegenerative disorders. This is a case study looking at Hunter syndrome, which is a lysosomal storage disorder that affects pediatric populations. It's normally treated, oh, sorry, this is actually not that slide. This is looking at neurofilament in Alzheimer's disease in one more clinical trial, but it is hot off the press. This one is a very recent study. In this case, Cassava is showing data from an ongoing open label study with simulfilam, which mechanistically supports altered filament A scaffolding protein. So mechanistically, this may be right in that sweet spot for what neurofilament is doing. And their interim results observed at 9 months that cognition and looked at cognition at 9 months and saw that lower NFL and other biomarkers were observed, and a 3-point benefit on the ADAS COG-11 scale was observed. So looking at multiple different biomarkers, of course, neurofilament isn't the one with the biggest effect, but it has a robust effect, and that's correlating at 9 months with other biomarkers and cognitive scales. So that's the last clinical study in age-related neurodegenerative disease. This is the one looking at a very recent trial presented just in late July. at the MPS symposium looking at Hunter syndrome. And this is a pediatric lysosomal storage disorder that's actually fairly well treated with enzyme replacement therapy, Elaprase, but that does not cross the blood-brain barrier. So the CNS effects of Hunter syndrome are still a big issue. Denali uses a transferrin receptor transport technology to deliver the drug across the BBB. And so this was a clinical trial to assess primary efficacy of that drug delivery system for Hunter syndrome. This is just showing interim 3-month data. They showed promising target engagement, but a concern in the sense that NFL levels were actually increased. Granted, this is a very small study, an N of 5, but the increase in NFL was observed across the small subcohort. I think what this emphasizes is, of course, the importance of monitoring other clinical scales and other biomarker outcomes. And also, I think it emphasizes the lack of normative NFL data in pediatric populations, because really knowing how NFL changes dynamically in pediatric disease is really a big unknown. So with that, we're just going to move on to the last little bit to position the questions for the panel. The discussions we've had so far talking about mechanism of neurofilament and the clinical trial data and case studies do position NFL as a high potential and robust biomarker for neurodegenerative disease. But depending on the disease, depending on the cohort, depending on what questions are being asked and what is known about the level of progression within the patients, I would say that classifying NFL as some of these different biomarker subgroups is really difficult to do. So for purposes of prognostic, I would say that there's robust data across multiple diseases that NFL is a very good prognostic biomarker. It may be a response biomarker for, say, MS. I think the jury's still out for some of the other diseases. It can possibly be predictive. Again, that's going to vary across the neurological disease. And because it isn't specific, it can be potentially used as a complementary diagnostic biomarker. But still, many questions remain around the heterogeneity and comorbidity of the subjects, the temporal complex relation to disease progression, the age of disease onset, potentially confounding age-related baseline effects, and then all of the other things that tend to haunt biomarker assays like sensitivity, cohorts, outcomes, normative data, standards, et cetera. So as we put all that together into kind of like a bucket of challenges, a bucket of potential solutions, I just try to capture some of the big picture ones here. Some of the big challenges remain standardizing sample collection and assay methods to align across multiple testing sites. This can, of course, be addressed by evaluating SOPs, standardizing and defining collection and storage process standards, calibrators, and also comparing assay methods. For the problem of lack of standardized normative databases in healthy control and disease subjects, this can be addressed by establishing the necessary inclusion-exclusionary parameters and identify the appropriate control groups. Again, making sure that this is over a broad age range, depending on what the disease is. There's an undetermined predictive value of NFL for disease progression and severity across neurodegenerative disease, but this can be addressed with cross-sectional validation in both early and advanced disease, and also in longitudinal studies. And for the challenge of fragmented data sharing in the repository ecosystem, this can be addressed by harmonizing data integration to access and utility and maximize utility. So as we kind of wrap it up and then switch over to the panel discussion, I want to leave you with some considerations for regulatory science as it applies to biomarkers, models, trials. I mean, all of these, are key considerations for leveraging biomarkers. In this particular context that we've focused today, a longitudinal biomarker is a type of measure with a dynamic behavior and variability. Any longitudinal measure, including a biomarker, must be understood dynamically within this context of the disease progression. And as we know, the disease progression and complexity and context from one neurodegenerative disease to another is very, very different and really complicated. Quantitatively understanding disease progression helps improve the understanding of biomarkers and other relevant sources of variability. And that such understanding can streamline the pathway towards regulatory acceptance of the drug development tools using biomarkers and models while improving clinical trial design efficiency. And as I mentioned, both assay validity and clinical validity need to be applied, and regulatory pathways exist for the review and potential acceptance of biomarkers within this context. So with that, I'm going to kick us off to our panel discussion. I am so thrilled today to have 3 experts joining us to help answer your questions and help us further understand what the key challenges and hurdles are in advancing the field of neurofilament as a biomarker for neurodegenerative disease. I want to thank Henrik Zetterberg, Matthis Synofzik, and Samantha Hutten. And apologies, Matthis, if I mispronounce your surname. We're going to switch now to an open panel session for Q&A in order to answer some of the questions that have been coming in. I know that Sadir and our CPATH team have been collecting the questions, so thank you for sharing them.
Yeah, thank you. Thank you very much, Terina. That was a really terrific overview of NFL in neurodegenerative diseases and both the basics as well as relevance to the clinical trials and drug development efforts. So I just want to welcome Henrik, Matthis, and Samantha. Thank you so much for your time and for joining us. And Henrik is one of my favorite person. We haven't met in person yet, but I've seen Henrik a lot of time on the video. So thank you so much, Henrik. We have a few questions coming in through the panel. So while our team is compiling those questions, I'll probably start with a couple of quick questions and maybe invite Klaus Romero, CSO, to provide clarity on a couple of points. So first, Henrik, I was just curious if you can just provide a brief background into one of your recent papers from JAMA Neurology in 2021, where you had kind of 3 aims. And one of the aims was to look at whether plasma p-tau 181 provides complementary information to non-disease specific plasma biomarkers of neurodegeneration, such as neurofilament light chain. So this was the January publication you had. And the conclusion of that study was that plasma NFL had pronounced associations with cognition and imaging markers of neurodegeneration. But in contrast to plasma p-tau 181, those associations were not AD specific. So the question I had for you, I know that since January, there's been much more studies going on. You published a lot more paper. Is there anything you can share with us as of today in terms of your understanding of the role of NFL, either as a complementary biomarker or on its own in terms of tracking disease progression with Alzheimer's disease as a case study, because you've been working in this space a lot?
Thank you, Sudir, for that introduction and for that question. I would just like to thank Terina for this summary on NFL. It's the most exhaustive and deep summary I've seen, I think. Very impressed. I'm actually a bit nervous to discuss NFL now. This paper where we compared the utility of phospho-tau in plasma with neurofilament light sort of highlighted what we hope will happen in the future in clinical chemistry, and that is that if you see a patient with cognitive problems, perhaps as a general practitioner, that one actually already then could take a neurofilament light and a phospho-tau test. And neurofilament light could then be regarded as sort of the troponin of the brain. We would have that as a general marker of neurodegeneration. We have to figure out reference limits carefully, of course. And then phospho-tau could tell whether there seems to be underlying Alzheimer's pathology contributing to the neurodegeneration. And if a person seeking medical advice is negative on both, perhaps then one should look into psychiatric diseases, depression, other causes of cognitive problems. Whereas if the patient is positive for NFL but negative for phospho-tau, perhaps then one should look into non-AD neurodegenerative dementias and prepare for a careful evaluation at the memory clinic or at the referral center. I don't think we have any new data that are adding to this reasoning. I think many research groups now have published papers that sort of at least perhaps this is my biased interpretation, but I think the field is moving towards supporting this use. Neurofilament light as a general marker, where you really have to figure out the cause of the underlying cause of the NFL increase, and phospho-tau as a very AD-specific biomarker, not positive in non-AD tauopathies, for example, almost like an amyloid response marker.
Yeah, thank you. Thank you very much, Henrik. And we are getting a lot of questions, so we're going to get to that one by one. So to kick it off, I want to probably loop in Klaus to one of the last slides that Terina showed in terms of regulatory considerations, So I was wondering if Klaus Romero, the Chief Science Officer who's on the call, Klaus, if you can provide a little bit of clarity on the point about the diagnostic utility of a biomarker versus its potential patient selection utility, and kind of put it in the context of what we are trying to do within CPAT as well as in terms of addressing the industry needs. Would you be able to provide some clarity on that?
Absolutely. Just quick check that you can hear me.
Yes, we can hear you, Klaus. Please go ahead.
Excellent, excellent. So yeah, I think this is right at the crux of several of the questions that were asked. And by the way, this is fantastic to see the different brilliant minds from the different consortia in neuroscience kind of coming together for this kind of topic. This is a great example of knowledge integration. So that's great. So in terms of quantitation, right? So let's take the term biomarker outside of the conversation a little bit, and let's understand that NFL is a measure of something, and that something is one of the still questions that are being researched right now. What does that represent in terms of mechanistic neurodegeneration, normal aging, etc.? So back to the question in the beginning, the framework in the beginning that Terina showed about the unknowns and the non-known knowns. So that's one component of this. And we need to continue to figure that out, and not just for NFL, but for any other measure that happens to be a measure that gets extracted from a biofluid. So that's one component. Then the other component is that you can also use these kinds of measures that are extracted from biosamples to make binary distinctions. Those binary distinctions can take the form of the D-word diagnosis, and that's great. But when you start going down that route, we all need to understand that relates to a specific type of medical product, likely a diagnostic device, an in vitro diagnostic device. That's great. But then that doesn't necessarily mean that just because you get a diagnostic device cleared by regulators, that automatically you understand everything that you need to understand in terms of how to deploy that to optimize patient selection approaches for clinical trials to measure the efficacy and safety of drugs. That's a different animal. And that's where the biomarker categorization that Terina showed is so critically important, because you don't necessarily need to go all the way to demonstrate the D-word diagnosis when you want to focus on optimizing patient selection for trials based on what the measure brings in terms of predicting what's going to happen with the disease or the drug effects later on. And that takes us to the longitudinal aspects. And Doug, as always, you're right on the money. The glimpses that we have right now, and that's what they are, glimpses of what the longitudinal utility of NFL can bring to the table is very promising. But we need to recognize that those are glimpses of what the actual longitudinal utility of NFL as a longitudinal measure can bring to the table. And the only way to figure that out is to integrate as many patient-level data sets as possible that have both the baseline and the longitudinal measure of NFL together with the clinical measures, and essentially quantify the heck out of the dynamics of the behavior of NFL and its linkage to the clinical metrics. That's the only way that we're going to start unraveling the question that still lingers: what is the actual value of NFL as a longitudinal metric? Does that help you track the disease at a very early stage where clinical manifestations are hard to come by? Does that, and that even if it's just that, it's of great value. Then there's the other questions. Does that help you track drug effects? And there was a question about ALS. I think that's another unknown. I would caution against drawing the conclusion that just because you don't see the change, that means that NFL doesn't track in a particular disease or for a particular drug effect. I think it's just a matter of us not having enough data and not having done extensive quantitation of the dynamics of those of the metric in that context to answer that question. So it's one of those beautiful examples of how everything comes together for data integration, data analytics, and the regulatory considerations. So I'll stop with that.
Yeah, thank you very much, Klaus. That's actually a good segue, because I want to give an opportunity for Matthis and Samantha, and of course Henrik as well, to see if they want to add anything to the question from Doug Langbein. Basically, I'll try to synthesize that, which is that most of the data, even for longitudinal studies, was actually for cross-sectional comparisons. But the usefulness of a measure of progression depends on repeated NFL measurements over time within the same patients. Can you comment on this distinction? I know that Klaus added a few comments there, but Matthis, Samantha, or Henrik, do you have anything else that you want to share with us from your expertise?
Yeah, thank you very much. So first of all, also my thanks to Terina. I think that was terrific. I couldn't have done such a great overview as Terina has been given. To the question in the chat, I think it is right that still many studies are cross-sectional, but the field is now really seeing more and more longitudinal studies from AD, but also from FTD, from ALS, and now also the first one from SCARS are coming up. So we are really getting more and more into the longitudinal field. And what we are seeing, and I think that comes to the core of our discussion, is that except for the conversion phase of those neurodegenerative diseases, in the, let's say, more moderate to advanced stages, the rate of NFL, they somewhat plateau off. They get to a stable rate. And what's important, and now coming back to the question, is even if, yes, we do need repeated measurements, we do need longitudinal measurements, however, we do not need to see an constantly increasing level of a certain biomarker. And that's a typical naive perspective on a biomarker, which very often in the NFL field takes place as well. You imagine that the more severe the disease, the higher the biomarker, and you need to find those correlations. And you don't find them in NFL, and you get disappointed about the power of NFL. But I think that's a wrong concept. Because I think the right concept, and I think that's the core of here, is that NFL does display not the severity of the disease, of the clinical disease, but it does reflect the neuronal cell turnover. And this means it goes up once the cells are decaying. And even if then the rate is up, this does not mean that the disease is stable, but the cell turnover rate is stable. And this is why it's not increasing anymore. Or maybe it's even going down in the advanced stages because the cell turnover rate is going down, maybe just because the crucial neurons are not there anymore. And I think so it's important to understand NFL not as a severity marker or progression marker in a classical sense, but as a cell turnover rate marker. And that concept, coming back to the question, is even if we have repeated time points and see that NFL is stable, it might still make a very good treatment response parameter, because if you then lower the increased cell turnover rate by your drug, this still means your drug is effective, because you lower the stably increased turnover rate gets reduced. So this means, yes, you need repeated measurements, but you do not need to see an increasing in your repeated measurements. Even lowering a stable measurement does speak in favor of treatment response of NFL.
Yeah, thanks, Matthis. Samantha, do you want to add anything there?
Sure. Thanks so much for having me, Terina and Sue here. Great to be here. I think along these lines, Terina highlighted some of the results from the PPMI study, so I won't reiterate, but despite the slow rate of change with disease progression that we did see in de novo PD, we're still excited because NFL is the first biochemical biomarker to increase over time and correlate with the motor and cognitive measures that we saw. So I think a critical next step in my mind that we haven't necessarily talked about yet is really around this combined modeling with other PD relevant biomarkers to better understand disease subtyping and progression, as we've talked about as an unmet need. So I'm a big proponent of the ETN framework that's being used in the Alzheimer's field now. And I imagine a future where we could have something like that for Parkinson's, where NFL is playing a big role. And to date, I think the data that we've seen thus far support a value for NFL and differential diagnosis and disease subtyping, as opposed to being useful for monitoring disease progression within the timeline of a clinical trial. But I think if we had a framework and NFL could be incorporated into that, as far as thinking about Parkinson's disease in the future, the way the Alzheimer's field has been moving towards, then some of these issues and questions could potentially be addressed.
Yeah, thanks so much, Samantha. So Henrik, if you can hold on to your comments, I think it's a segue to go to the next question, because I want to make sure we have a chance to address, which is actually, I have Bob Alexander's question: what does the increase in NFL in normal aging reflect? And I think this is kind of related to what Matthis was talking about, how NFL fundamentally reflects neuronal turnover. So normal aging, we do have the process. Would you like to start with that, Henrik, and then we can just connect the topics in?
Yeah, I think it actually reflects this well-known loss of brain volume with age, the normal loss of brain volume. And it's very clear that there are two phases in the NFL increase. So if you look, we have data now from 5 years to 95, and throughout life there is a small increase with age, which is it's very minor, but it's clear and significant. But then from 50, 60 years of age, the increase takes off a bit, and you see also that some individuals get high levels that are more than what you would have projected from the age-related increase with time, with age-related decrease over the lifespan, sort of. And those we do not know that well what these individuals have, because of course they were sampled as healthy controls. But I think it would be exciting to use to do a longitudinal follow-up and see what happens to these people, because perhaps they have something going on that will eventually present clinically, or they have something minor that happened that gave a peak in NFL, and then it might not deteriorate further. But I think there is something with the age-related increase which has to do with brain volume changes that are happening with age. And then one could question if that is normal or not. But to some extent, I think most think that aging comes with some loss of brain volume.
Yeah, thanks, Henrik. So Samantha and Matthis, I'm going to ask you to hold off to your comments first, because Enchil Liu has a very important question that's very, very relevant for everything that we do here. But before we go there, I want to pull up Jeremy Edgerton's question, which is that the hypothesis is NFL in CSF and serum represents release of the protein from degenerating neurons. But could disease-related changes in protein synthesis or clearance rates also contribute? What do we know about NFL production and clearance in neurodegenerative diseases? So I think this has some relevance to what you are already talking about, Matthis. So if you want to start off with that, we'll then go to Samantha and then come to Enchil Liu's important question, if that sounds good.
So I think we still need more research on both sides, on the clearance and the production, but maybe start with the clearance. So it seems that renal function, at least some studies point to the effect that renal function does have an effect. At least they show correlation between NFL and creatinine. Some of them are not very well controlled for age. So you still, of course, because of aging, it does say to reduce your renal function, it does increase your NFL. So I still don't think those are the best studies ever, but there seems to be an effect on renal function. That's one of the clearance effect. I'm not aware of any others, but maybe Henrik can can comment on this in a moment. What is really important or interesting, however, is that even though we are all studying NFL across all neurodegenerative diseases, we are not very sure about the exact release mechanisms. And is it only a passive release? Does it also have an active component? Is it both? And how does it release during neuronal development? How is it released during neurodegeneration? So it's really remarkable that we all keep talking about the NFL like tonight, and we don't really understand the release mechanisms. And we are now in our lab, we are now going back really to stem cell work and really trying to go do it on a single cell level and try to find out more about the release mechanisms. But this is still largely pending in the field. And this, Jeremy, without this, we don't really know which mechanisms, let alone which protein aggregation in neurodegenerative diseases do drive the NFL increase. Is it really only axonal loss? Or does, for example, the membrane already get permeable at certain stages, even before the axon is damaged per se? That, at least to my knowledge, we don't know.
Thanks, Matthis. Samantha, any quick comment there?
No, I think you've covered it. I think there's a lot of biology that we still need to understand before we can continue to probe some of these biomarker questions, but hopefully all that is happening in parallel.
Okay, thanks, Samantha. So let's move on to Enchi Liu's question, which is a very important question. Claude, so if you can please stand by to respond and provide some context. So Enchi asked, is there a utility to ban across neurodegenerative diseases to qualify NFL for a specific context of use? Especially useful would be disease monitoring, which is a jumping off point for use in therapeutic trials for response monitoring. So, Klaus, would you like to comment on that and provide some context based on the work we do, which I think is very relevant to us as well?
Sure. Yeah, absolutely. That's a relevant question. So, I would flip the question a little bit and say, okay, so how can we together through the public-private partnership environment that we can provide through the different consortia work in a collaborative manner to generate the necessary scientific evidence to generate the confidence that is needed for NFL to potentially be deployed as a relevant biomarker in clinical trials to capture the efficacy and safety of potential new drugs for the different diseases that we're working on. Then if that's a goal to generate that solid foundation so that sponsors can have the confidence to deploy the biomarker in the clinical trials, then what are the mechanisms to make that happen? And so the mechanisms are several, not just qualification. Qualification is one of several mechanisms by which you can reach that objective. But at this point in time, I would concentrate on honing in on providing that intended application, aka what you rightfully so call the context of use. The context of use not just for the biomarker as a biomarker, but the quantitative machinery that you need behind the biomarker in order for sponsors to really understand how to best deploy it in clinical trials. So the answer is yes. Let's aim towards the goal of generating that confidence that is needed. And in the process of doing so, we can identify the best regulatory pathways to follow.
Yeah, thanks. Thank you so much, Klaus. So we have another 11 minutes to go, and I have some couple of quick important questions come through. I want to make sure that we at least address those. The next quick one is from Jordi, and probably if Brian Gordon is on the call, I know that he put in a comment, probably this is relevant to him. So Jordi's question was, in clinical trials using second generation antibodies to treat AD, where all of them clear A beta, what is the overall behavior of NFL levels? So Brian Gordon, if you're on the call, I know that you kind of provided a very relevant comment actually earlier on the WebEx chat. Would you like to comment on this question based on your own experience, from the gantenerumab, solanezumab studies and some of the results that you just published in June?
Sure, and I put a link in the chat to the paper that just came out reporting the results from the Diane readout, which had SOLA and GANT in it. And for exactly this point, where in the GANT arm, we saw a reduction in amyloid, but we also saw a slowing in the rate of change of NFL. And our interpretation of that is that it's indicative of a downstream effect of clearing amyloid on neurodegeneration. And the NFL actually was more sensitive than MRI and FDG, which you can imagine might take some time to accrue, versus that NFL is a very nice kind of short-term marker of what's happening to neurodegeneration right now in the system. And you see this slowing on people on GANT, and it was relatively proportional to the amyloid change.
All right. Okay. Thank you, Brian. So I know that Lars had a question. I'm going to hold off on that, and we'll come to that in a minute or two. But I want to go back to the question from Milton Biagioni, which is that as NFL is not linked to any specific pathology and can rise years before phenotype conversion, it's very challenging to use it as subtyping or enrichment. We likely need to enrich NFL by exclusion of co-pathology. What biomarker approach could be envisioned in the PD space, MRI, tau, amyloid, enrich NFL to exclude AD and vascular dementia, and that could be a critical, for example. So do anyone in the panel, so Henrik, Matthis, or Samantha, would you like to comment on that question from Milton?
I could just start briefly and state that I think there could be a use in, if you want a pure Parkinson cohort with no Alzheimer comorbidity, I think one could make use of phospho-tau again there. and exclude patients with increased levels of phospho-tau in the clinical trial. Neurofilament light also has some use in this Parkinsonian disorder context because the atypical Parkinsonian disorders have increased levels, whereas pure PD typically don't. And I think Samantha's work together with Britt Mollenhauer and other researchers also underscore that if NFL is increased one, there is additional neurodegeneration going on, which is not just linked to the early neurodegeneration in substantia nigra associated brain regions. So it's I think both phospho-tau and neurofilament light could be useful in this aspect. The plasma A-beta biomarkers, they are difficult because the fold change between amyloid positive and negative people in A-beta 42/40 ratio is so small in plasma compared with CSF, and that is because this change happens on the top of peripheral beta amyloid. So I would advise on using, again, phospho-tau and neurofilament light as a pair.
I agree, and I think those data sets are available in PPMI and other cohorts. And to the question earlier about what we can be doing across these different diseases, I think the more that we can measure sort of the non-traditional analytes, so in Parkinson's disease, looking at things like NFL, A-beta, tau, phospho-tau, and in some of the other cohorts, including things like alpha-synuclein, I think that will help us to parse out some of these questions, especially as Terina noted, because even though we recruited for a Parkinson's disease cohort, we did end up with subjects who ended up being other neurodegenerative disease categories, and that can be informative in each of our longitudinal cohort studies as well.
I could just add quickly also and state that I've been so excited by the RT-QuIC results on alpha-synuclein. I mean, you have to have a lumbar CSF sample then, but it looks really promising as a qualitative biomarker demonstrating whether or not there is alpha-synuclein pathology. And this has been validated now in autopsy studies in a way that looks much better than I could ever have dreamt of.
Thank you. Thank you so much, Henrik and Samantha. So stand by it. So I'm going to try to combine the two other questions we have before coming back to Doug Langbaum's comment earlier on, which is the question from David Levlin and Lars. So David asked, to what extent is NFL important predictively in isolation when compared to a more multimodal approach? It's also not clear the degree to which simple categories or linear combinations of biomarkers will be sufficient versus machine learning approaches that allow us to model complexity and interactions more effectively. I think, again, Lars, correct me if I'm wrong, but you had a kind of a related question from a very high level, which is that the true utility of NFL in AD, for example, the most of the diagnostic value disappears when adjusting for something like a patient's MMSE score, which is arguably easier to measure. So what are the steps the field must take to begin addressing the real utility and added value of NFL in a clinical setting? So from the Critical Path Institute perspective, when we do the modeling and analysis, that's one key area we focus on. How does individual biomarkers change over time, but also how does an individual biomarker correlate with other assessments that we have, either other biomarkers or cognitive assessments? So I wonder if anyone in the panel, Henrik, I think you have done some modeling, but Matthis, Samantha, if you have anything to comment, but this is an area where probably Klaus can provide some context based on what we do within the Critical Path Institute as well. So Henrik, would you like to start off with some brief response to that?
Yeah, we have done quite little modeling, I have to say, but instead we often look at the biomarkers in isolation, which is a bit old-fashioned. I know that, but and then I would almost think about how to use NFL in a clinical trial as one measure of neurodegeneration to evaluate in relation to treatment arm and doses and compare, like we often do, placebo versus active treatment in different doses. And then I think the hope is that the different biomarkers we have at hand, if one directly should put up two neurodegeneration markers, MRI and neurofilament light, then perhaps neurofilament light could predict future MRI changes or something like that and be a little bit faster than. But then one has to remember also that the half-life of NFL in biofluids is like 2-3 months almost after acute injury. So it's not super fast as a biomarker. Whereas other markers like GFAP and so have a much shorter half-life. So I think we should try to learn as much as possible on each individual biomarker And the discussion before is relevant to this also, production and clearance rates, where we need to learn more, and where neurofilament light has been particularly challenging. Randy Bateman's team has tried to do silk and determine production and clearance rates, but it's so slow, and the levels are so relatively low, so it's challenging, but I know they haven't given up.
And maybe if I
Thanks,
can
Henrik.
add,
Go ahead, Matthis.
maybe if we can add to this, I think the main value of NFL in neurodegenerative diseases is not so much its diagnostic value. I think it has there, and maybe prion disease, maybe ALS, but otherwise the overlap is so strong. But for example, for all the genetic ones, we don't need this, and maybe even for the sporadic ones, we might have alpha-synuclein and so on. So maybe at the long term, we don't even need diagnostic. We might have better ones than NFL. So the power will not be NFL. The power is really the reflection of axonal degeneration. And what is important is, and that's your question, Lars, for example, what is the advantage compared to the MMSE or something? The good thing about NFL, it's so stable that you can really reduce your sample sizes by adding it to your outcome measures. And this is, we have, I think, Henrik, you have done it in your Huntington paper, we have done it in our SCAR3 paper, now we have an FDD paper under review where we do sample size calculations based on NFL reduction. And given the stability, the short-term stability of the NFL measurements, you can really, as compared, for example, to a clinical scale, may it be the MSE or whatever, which are so variable, which you really lead to large cohorts. With such a stable marker than NFL, you can really reduce the sample sizes if you want to measure treatment response. And I think that's one of the powers where we might need NFL.
Thank you, Henrik. Samantha, do you want to add anything as kind of a closing comment there?
Just to say, I think oftentimes we focus on these individual analyses, but more and more now that we have these big data sets coming through studies like BPMI and also the Accelerated Medicines Partnership or AMP-PD effort, we are focused on modeling, and we do have some open requests for funding now, specifically around data analysis and multimodal machine learning algorithms, so that we can really try and parse this out, because this is a critical need for where we are right now with these data sets.
Yeah, thank you. Thank you so much, Samantha. So we have 2 minutes left. So I want to probably bring back Doug's question, but maybe for Henrik, because you know, you're kind of leading a lot of this analytical validity and assays. And I've been part of some of the recent presentations you've had at the GBSE as well, Henrik. So the Doug's follow-up question to his earlier comment was that, you know, just talking about the longitudinal stability of the assay readouts, assuming that there is a true biological steady state in the disease when it comes to, you know, looking at NFL biomarker assays. Is there anything you can share in terms of, I know that there's a lot of work that's ongoing in your group and in that GBSE. Is there anything you can share with us that will provide some idea in the next, let's say, 60 seconds?
I could just quickly mention that in the GBSC, the Global Biomarker Standardization Consortium, we have just presented some assay comparison results and showing these very nice correlations for NFL across analytical platforms, and many of these still use the human diagnostics antibody pair, but there are some studies that don't, and they still correlate. The trick is to focus on the rod or core domain of the protein and avoid the N and C termini. That's very important because they are probably processed and proteolytically degraded. So the trick is to go for the mid domain of the protein. Many of the big chemistry companies are actively looking into NFL as a biomarker. And I am pretty sure that we will see from companies like Abbott and Roche and additional players in this field, together with Quanterix and other and Mesoscale Discovery and those companies, that we will see a number of automated tests coming up now. And we are working also on new commutability studies to see if we can also create reference materials so that we eventually could calibrate these different assays to each other and achieve some standardization in this field. So I think this will work out well, actually. It's given the nice correlations we see.
Yeah, thanks. That's absolutely true. And that was a very important point you mentioned towards the very end, Henrik, nice correlations we see. And that's something CPAT we are focusing on within all the 5 Neuroscience Consortium, and all of you are aware of that. As part of our modeling and analysis work, we are aiming to targeting more data sets, more contemporary data sets, rich in biomarkers, mainly for generating those clinically meaningful correlations and associations as we do the disease progression modeling from our end. So I know that we are at the top of the hour. So thank you so much to everyone, particularly Henrik, Matthis, Samantha, and everyone who participated in this webinar today. This was a tremendous success. But please know that this is not the end of this conversation. As you all know, we will be bringing this to the individual consortium coordinating committee, modeling working group calls as we go forward. And this will be a key topic that we'll be working on, driven by patient level data and also the extremely hard work that we are doing on our end to incorporate those key biomarkers into our modeling and analysis plans, driven by the feedback that we get from you on a regular basis. So thank you so much to everyone for this really terrific webinar. I'm very happy that this was super good. So once again, I appreciate your time. And Henrik, Matthis, Samantha, thank you so much for your support and partnership and collaboration and for joining us today. So everyone, wish you all a good rest of the day, and we will talk to you soon in the upcoming consortia specific meetings. Thank you, everyone.
Thanks.
Thank you very much.
Bye-bye.